Welcome back, friends. Welcome back again to one more other lecture session from this channel. So in this lecture session, we will be studying regarding tools of recombinant DNA technology from biotechnology principles and processes in detail. So here, as we have already studied, as we have already learned in our earlier lecture session, basically recombinant DNA technology, it takes the advantage of various key tools. So here the various key tools which are required for recombinant DNA technology, they may include restriction enzymes, polymerases, ligases, vectors and host organisms. So here all these are the various key tools which are required for organizing recombinant DNA technology. Is it clear? So let me write it on the board. Recombinant DNA technology. Recombinant DNA technology takes the advantage of various key tools and these may include restriction enzymes, polymerases, ligases, vector and host organism. So here all these are the various key tools which are required for organizing recombinant DNA technology. So right now we will be studying regarding each and every key tool in detail. So at first if we consider restriction enzymes. So here what are restriction enzymes here? As the name itself suggests, these are the specific enzymes which are responsible for cutting only a restricted part of the DNA fragment from the desirable source. So the reason why these specific enzymes, they are termed as restriction enzymes. Is it clear? So let me write it on the board. Restriction enzymes. These are the specific enzymes. Which are responsible for cutting only the restricted part of DNA fragment. DNA fragment from the desirable source. Right? So such specific enzymes, they are termed as what? Restriction enzymes. So here for the very first time, these restriction enzymes, they were identified in the year 1963. So here the particular restriction enzymes, most commonly they are responsible for restricting the growth of bacteriophage in E. coli host cell. Is it clear? The specific enzymes, they are responsible for restricting the growth of bacteriophage in E. coli host cell. So let me write it here. In the year 1963, restriction enzymes were identified. So these enzymes are responsible for restricting the growth of bacteriophage the growth of bacteriophage in E. coli host cell. In E. coli host cell, right? Is it clear? As we all know, bacteriophages are the viruses which take the advantage of E. coli as the host cell, right? So here these specific enzymes, they were responsible for restricting the growth of bacteriophage in the E. coli host cell. And here the specific enzymes which were identified for the very first time, they were responsible for recruiting the methyl group to the DNA, and even they are also responsible for cutting the DNA fragment. Is it clear? The specific enzymes. So let me write it here. Specific enzymes which were identified for the very first time they are responsible for recruiting methyl group to the DNA sequence. That means these particular enzymes, they are responsible for recruiting or for adding the methyl group to the DNA sequence. And even the other enzyme, it is responsible for cleaving or for cutting the DNA fragment into multiple pieces. Is it clear? Methyl group to the DNA sequence. And the other enzyme is responsible for cutting the DNA fragment. DNA fragment at specific site. So here the very first restriction enzyme which was identified, here it was nothing but what? Hind2. So here Hind2 restriction enzyme, it was capable to recognize the particular DNA sequence by taking the advantage of a 6 base pair sequence which is termed as recognition sequence. Is it clear? The very first restriction enzyme which was identified, here it was nothing but Hind2 restriction enzyme. And here it was came to know that the Hind2 restriction enzyme, it was capable to recognize, or it is capable to recognize the DNA sequence by taking the advantage of a 6 base pair sequence which is termed as recognition site or recognition sequence. So here by taking the advantage of a 6 base pair sequence which is termed as recognition site or recognition sequence, the Hind2 restriction enzyme, it is capable to recognize the DNA sequence. Is it clear? Hind2 was the first restriction enzyme which was identified. And it was came to know that Hind2 restriction enzyme takes the advantage of 6 base pair sequence. So here the 6 base pair sequence, it acts as recognition site or recognition sequence for the particular concerned enzyme. So here by taking the advantage of the 6 base pair sequence, which is termed as recognition site or recognition sequence, the Hind2 restriction enzyme, it is capable to recognize the DNA sequence. So here after the successful discovery of the Hind2 restriction enzyme, then after many of the restriction enzymes they were successfully identified. And here it was came to know that till today nearly 900 restriction enzymes they were identified from more than 230 bacterial strains. So here nearly 900 restriction enzymes they were identified or they were isolated from nearly 230 bacterial strains. Is it clear? So let me arrange the things here. So here, as already mentioned, nearly 900 restriction enzymes they were identified or they were isolated from nearly 230 bacterial strains. And here each and every restriction enzyme they are very site specific. By taking the advantage of a specific base pair sequence termed as recognition site or recognition sequence, each and every restriction enzyme they are capable to recognize the concerned DNA sequence. Is it clear? So let me write it here. Nearly 900 restriction enzymes were identified and isolated from 230 bacterial strains. And each and every restriction enzyme they are very site specific. By taking the advantage of a specific base pair sequence termed as recognition site or recognition sequence, each and every restriction enzyme they are capable to recognize the concerned DNA sequence. Each and every restriction enzyme are site specific. And here, if you look at the nomenclature of the restriction enzyme, so here each and every restriction enzyme it is having one concerned name. So here in order to induce or in order to provide one concerned name to the restriction enzyme, there are certain strategies which were taken into the consideration by the nomenclature of the restriction enzyme. And these strategies they may include name of the genus, name of the species of the concerned microbe from which the restriction enzyme is isolated, and the other specific strategy is nothing but whether the particular strain is smooth strain or rough strain, and the other strategy is nothing but the Roman number. Is it clear? So let me write it here. The nomenclature of restriction enzymes had taken certain strategies into consideration. For imparting or providing one concerned name to the restriction enzyme. And these specific strategies they may include name of the genus, and the other one is name of the species of the concerned microbe from which the restriction enzyme is isolated, and the other one is Roman number Roman number, and the other specific strategy is whether the concerned microbial strain it is a smooth strain or rough strain. Is it clear? Smooth or rough strain. So here, by taking the advantage of all these strategies into consideration, one concerned name is given to the restriction enzyme. So here, in order to understand this particular concept, here we can take the advantage of one common example which has mentioned in your NCERT textbook. So if we consider the eco R1, so let me write it here, eco R1. So in case of eco R1, which is the concerned restriction enzyme, so here the particular eco R1, in case of eco R1, here E stands for Escherichia, which is nothing but the genus name. Is it clear? Escherichia, which is nothing but the genus name. Genus name. And here Co stands for coli, which is nothing but the species name. And here R stands for rough strain. That means the concerned restriction enzyme, it is isolated or it is identified from the rough strain. Rough strain. And here 1 stands for Roman number. So here the concerned Roman number, it indicates the order of the particular concerned restriction enzyme which is isolated from the particular microbial strain. Is it clear? So once when we intermingle all these things, here it is together termed as what? Eco R1. Is it clear? So in this way, by taking the advantage of all these strategies into consideration, each and every restriction enzyme, it has acquired one concerned name from the nomenclature of the restriction enzymes. Is it clear? And here basically restriction enzymes, they are capable to cleave nucleic acids. So the reason why restriction enzymes, they belong to a specific class termed as nucleases. Is it clear? So let me write it here. Restriction enzymes. Restriction enzymes are capable to cleave the nucleic acid sequence. Nucleic acid sequence. So the reason why these are called as nucleases, these are called as nucleases. So here the concerned nucleases, basically they can be classified into two different types, and these may include exonucleases and the other one is endonucleases. So simply basing on the type of the action, here we can classify the concerned nucleases into two different types, and these may include exonucleases and the other one is endonucleases. These nucleases are of two different types. Two different types which may include exonuclease and the other one is endonuclease. So here most commonly exonuclease, as the name itself suggests, these are the specific enzymes which most commonly target the extreme ends of the nucleic acid sequence. But whereas endonucleases, most commonly they are responsible for targeting the internal portion of the nucleic acid sequence. So the reason why they are termed as endonucleases. Is it clear? So let me arrange the things here. Here, as already mentioned, the specific nucleases, basically they are of two different types, and these may include exonucleases and endonucleases. So here exonucleases, most commonly they target the extreme ends of the DNA sequence, which may include extreme 5' end and even the extreme 3' end. But whereas endonucleases, they are capable to target the internal portions of the DNA sequence. So the reason why they are termed as endonucleases. Is it clear? So let me write it here. Exonucleases are capable to target the extreme ends of the DNA sequence. Extreme ends of the DNA sequence. Extreme ends of the DNA sequence. Either it may be the extreme 5' end and even the extreme 3' end. But whereas endonucleases, they are capable to target the internal portions of the DNA sequence. Endonucleases are capable to target the internal portions of the DNA sequence. The internal portions of the DNA sequence. Internal portions of the DNA sequence. So here the specific restriction enzymes, which may include exonucleases and even the endonucleases, they are capable to recognize the concerned DNA sequence by taking the advantage of a specific base pair sequence termed as recognition sequence. So here, by taking the advantage of the particular recognition sequence, each and every restriction enzyme, they are capable to recognize the concerned DNA sequence, because basically all the restriction enzymes, they are very site specific. So here each and every restriction enzyme, they are capable to recognize the DNA sequence only by taking the advantage of recognition sequence or recognition site. So here once when the restriction enzyme, once when it recognizes the recognition sequence, the concerned restriction enzyme, they are capable to cleave the sugar and the phosphate backbone which are present on both the single strands of the double-stranded DNA. Is it clear? Restriction enzymes are site-specific. Site-specific. These enzymes are capable to recognize the DNA sequence by taking the advantage of recognition sequence. Recognition sequence, right? So here once when the restriction enzyme, once when it recognizes the recognition sequence, these enzymes, they are capable to cleave the sugar and the phosphate backbone which is present on both the single strands of the double-stranded DNA. Is it clear? Restriction enzymes are capable to target the DNA sequence by cleaving the sugar and the phosphate backbone. The sugar and phosphate backbone which are present on both the single strands of double-stranded DNA. On both single strands of double-stranded DNA. Single strands of double-stranded DNA. So here the specific restriction enzyme, it is capable to inspect the DNA sequence by taking the advantage of recognition sequence. So here the recognition sequence, basically it is of palindromic site. Is it clear? The particular restriction enzyme, it is capable to inspect the particular DNA sequence by taking the advantage of recognition site or recognition sequence. So here basically the recognition site or recognition sequence, it is palindromic in nature. So here what is the meaning of palindrome here? So here in order to understand what is palindromic sequence, here we can take the advantage of many of the common examples. Is it clear? Restriction enzymes. Restriction enzymes are capable to target the recognition sequence which is palindromic in nature. Palindromic in nature. Palindromic in nature. So here, in order to understand what is the meaning of palindromic nature, here we can take the advantage of many of the common examples. So if we consider some of the words like madam, right? Madam, rotor, and Malayalam, right? So in case of all these three words, so here all these three words, they are having similar sequential order of alphabets when we read from front to back and from back to front. So if we consider madam, so here from front to back it is madam, and even from back to front it is madam. Is it clear? Then in the same way, if we consider rotor, so here from front to back it is rotor, and even from back to front it is also rotor. And even if we consider Malayalam, from front to back it is Malayalam, and even from back to front it is also Malayalam. So such specific words, they are termed as what? Palindromic words. And if we apply the same concept to the base pair sequence which is present in case of the DNA, so such specific base pair sequence, they are termed as what? Palindromic sequences. Is it clear? Palindromic sequences. Palindromic sequences are responsible for the recognition by the restriction enzymes. And these base pair sequence has similar base pairs from front to back, and even from back to front, So such specific sequences or such specific base pair sequences, they are termed as what? Palindromic sequences. Is it clear? So let me arrange the things here. So here, in order to understand the nature of the palindromic sequence, here we can take the advantage of one of the common example which has mentioned in your NCERT. So if we consider 5' GAA TTC 3', and here it is 3' CTT AAG 5'. So here from 5' to 3', it is GAA TTC 3', and from 3' to 5', it is CTT AAG 5'. So such specific sequences, they are termed as what? Palindromic sequences. So here by taking the advantage of palindromic sequence, which is a part of the recognition sequence, each and every restriction enzyme, they are capable to recognize the DNA sequence here. So here we need to understand that though the specific restriction enzymes, they are very site specific, but the particular restriction enzymes, they won't cleave accurately at the concerned recognition sequence. The particular restriction enzymes, they are capable to recognize the recognition sequence by leaving a short gap at the palindromic site. Is it clear? Restriction enzymes are capable to recognize the recognition sequence by leaving a short gap at the palindromic site. So here, because of leaving a short gap at the concerned palindromic site, it leads to the exposure of overhangings of the short stretch of the DNA sequence. So here the overhangings of the short stretch of the DNA sequence, here it is termed as sticky ends. So here the concerned sticky ends, they act as hotspots for the successful binding of complementary countercut parts. Is it clear? Because of the exposure of overhangings of the short stretch of the DNA sequence, DNA sequence termed as sticky ends, the complementary countercut parts, countercut parts are capable to associate here. Is it clear? So here after the successful association of the complementary countercut parts, here the native DNA in association with the foreign DNA, it is capable to remain in association by taking the advantage of a specific enzyme termed as ligase. So here ligase is the specific enzyme which is responsible for inducing the ligation. So here once when the particular ligase enzyme, once when it induces the ligation, here it leads to the formation of recombinant DNA. Is it clear? So let me write it here. After the successful association of the foreign DNA with the native DNA, the concerned foreign DNA may remain in association with the native DNA by taking the advantage of ligase, which induces ligation, which induces ligation, which induces ligation. So here, after the successful ligation of the foreign DNA with the native DNA, here it leads to the formation of recombinant DNA. Is it clear? So here, in order to understand this particular concept, here we can take the advantage of one of the diagrammatic presentation which has mentioned in your NCERT. Is it clear? So let me arrange the things here. So as already mentioned, the specific restriction enzymes, they are capable to recognize the concerned recognition sequence by leaving a short gap at the concerned palindromic site. So if you look at here, the cleavage of the DNA sequence, it has took place by leaving a short gap at the concerned palindromic site. Then in the same way, here the cleavage of the DNA sequence, it has took place by leaving a short gap at the concerned palindromic site. So here, after the successful cleavage of the concerned DNA sequence by taking the advantage of the specific restriction enzyme, now here both the sources of the DNA, they are capable to undergo ligation by taking the advantage of a specific enzyme termed as ligase. So here, once when the specific ligase enzyme, once when it induces the ligation, now here it leads to the formation of a specific DNA termed as recombinant DNA. So here, recombinant DNA, it is an association of two different DNAs which were obtained from two different sources. Is it clear? So here, because of the presence of the unique nature of the concerned restriction enzymes, so here most commonly restriction enzymes, they are used in case of genetic engineering or in case of rDNA technology in order to construct recombinant DNA molecules. Is it clear? So let me write it here. Because of the unique nature of restriction enzymes, in constructing the recombinant DNA molecules, these enzymes are used in recombinant DNA technology. And here we need to understand that the specific restriction enzyme which is most commonly used in order to cut the foreign DNA, such specific restriction enzymes, even they are used in order to cut the vector DNA molecules in order to construct the recombinant DNA. Is it clear? The specific restriction enzyme which is most commonly used in order to cut the foreign DNA, the same restriction enzyme, even it is also used in order to cut the vector DNA molecules in order to form a recombinant DNA. So let me write it here. Specific restriction enzyme which is most commonly used, which is mostly used in cutting the foreign DNA, the specific restriction enzyme, even it is also used in cutting the vector DNA molecules, vector DNA molecules. Is it clear? So then after we will be studying regarding separation and isolation of DNA fragments by taking the advantage of gel electrophoresis in detail. Is it clear? So let me arrange the things here. So here, after the successful formation of recombinant DNA by taking the advantage of restriction enzymes and even the DNA ligase, now here the recombinant DNA plasmid, it is inserted into the E. coli host. Now here likewise the other genes of the plasmid DNA, even the foreign DNA which has many number of genes, they are capable to undergo expression within the E. coli host cell. And here the specific mechanism or the specific process, here it is termed as gene cloning or gene transfer. Is it clear? And then after here we will be studying regarding separation and isolation of DNA fragments. So here after the successful isolation of the DNA from the concerned desirable source, now here the particular DNA sequence, it is cut into multiple fragments by taking the advantage of a specific enzyme termed as restriction enzymes. Is it clear? So let me write it here. After the successful isolation of DNA from the source, the DNA sequence, it is cut into multiple fragments. By the means of restriction enzymes. Now here the particular multiple fragments, they are successfully inserted into the electrophoretic chamber by taking the advantage of the specific wells here. Is it clear? The specific DNA fragments which are successfully cut into multiple fragments by taking the advantage of restriction enzymes. Now here these specific fragments, they are successfully inserted into the electrophoretic chamber by taking the advantage of wells here. So here after the successful placement of the multiple fragments into the electrophoretic chamber by taking the advantage of wells, now here the specific DNA fragments, they are negatively charged, they are capable to move towards the anode. Is it clear? As the specific DNA fragments, they are negatively charged, they are capable to move towards the anode by the means of a specific medium or by taking the advantage of a specific matrix which is composed of agarose gel. Is it clear? So let me write it here. Specific DNA fragments are placed into the electrophoretic chamber by the means of wells. So here DNA as it is negatively charged, it is capable to move towards the anode which is positively charged through a specific matrix made up of agarose gel. made up of agarose gel. Is it clear? So here the specific DNA, as it is negatively charged, it is capable to move towards the anode. And here we need to understand that the larger DNA fragments, they get settled at the top. But whereas the smaller DNA fragments, they are capable to move through the agarose gel by the means of a specific effect termed as sieving effect. Is it clear? The smaller DNA fragments, they are capable to move through the agarose gel by the means of a specific effect termed as sieving effect. So let me write it here. Larger DNA fragments are capable to settle at the top, but whereas the smaller DNA fragments are capable to move through the agarose gel by the means of sieving effect. So here the meaning of sieving effect is very simple. So here the particular agarose gel, it acts as sieve pore-like structures. Is it clear? The particular agarose gel, it has many number of sieve pore-like structures. So here these sieve pore-like structures which are present in case of the agarose gel, they are capable to allow only the smaller DNA fragments. So the reason why the smaller DNA fragments, they are capable to travel longer in comparison with the larger fragments. The larger DNA fragments, they are capable to settle at the top, but whereas the smaller DNA fragments, because of the presence of a specific effect termed as sieving effect within the agarose gel, here the smaller DNA fragments, they are capable to settle at the bottom, but whereas the larger DNA fragments, they are capable to settle at the top. So here after the successful migration of the DNA fragments towards the anode, now here the specific DNA fragments, they are strained by taking the advantage of a specific dye termed as ethidium bromide. Is it clear? The concerned DNA fragments, now here they are successfully strained by taking the advantage of a specific dye termed as ethidium bromide. So let me write it here. So let me arrange the things here. The DNA fragments so let me write it here. DNA fragments, specific DNA fragments are stained by the means of specific dye like ethidium bromide. So here after staining the specific DNA fragments by taking the advantage of a specific dye like ethidium bromide, now here the entire electrophoretic apparatus, it is successfully exposed to the UV radiation by taking the advantage of a specific device termed as transilluminator. So here, once when the specific stained DNA fragments, once when they are exposed to the UV radiation by taking the advantage of a specific device termed as transilluminator, now here it leads to the exposure of orange colored DNA fragments. Is it clear? Once when the specific stained DNA fragments, once when they are successfully exposed to the UV radiation by taking the advantage of a specific device termed as transilluminator, it leads to the exposure of, or it leads to the formation of a specific orange colored DNA fragments. So here these orange colored DNA fragments, they are successfully isolated or they are successfully cleaved from the agarose gel by the means of a specific procedure termed as elution. Is it clear? DNA fragments are isolated from the agarose gel by the means of elution. So here, after the successful isolation of the concerned DNA fragments from the particular electrophoretic chamber, now here the specific DNA fragments, most commonly they are aligned with the native plasmid DNA by taking the advantage of a specific enzyme termed as DNA ligase. So here the specific DNA ligase, it is responsible for inducing the ligation in between the specific DNA fragment and even in between the native plasmid DNA in order to form the specific recombinant DNA molecules. Is it clear? So here this is regarding the tools of recombinant DNA technology from biotechnology principles and processes in detail. And in the next upcoming lecture session, we will be studying regarding cloning vectors in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.